I'm going to jump directly to it, and I'm representing the Links Data Coordination Center. And we have some really exciting new ways to visualize the Links data. So for those of you who don't know, one of the most exciting parts of Links is that we are collecting data in high throughput for many perturbations applied to human cell lines, and then measuring a lot of things like transcriptomics, proteomics, cell viability, and other types of data. So in here, what you're looking at is visualization of 2,344 gene expression signatures. So a signature is when you compare controlled, unperturbed cells. Here it's like 6 breast cancer cell lines treated with 105 drugs, and those drugs were applied in 6 different doses, and gene expression was measured at 2 time points. So you can see that the perturbations are clustered into some typical modes of action, and in green those are cell cycle drugs. So a lot of the cell cycle drugs are doing basically the same thing to those cells, and there are some areas that, so basically the responses are clustered by the drug class. On the right, so this is a collaboration between the Broad Transcriptomics Center, the HMS Link Center, and we've contributed this type of visualization. It's an interactive website that you can go to. And on the right, you have the cell viability measured for those cells. So you can see that in blue are the cells that are dying due to the drug treatment, and in red, you actually see an increase in proliferation of those cells. So some cell types, cell lines, for some of those drugs, they escape the killing and they actually increase in their proliferation. So I'm just going to show you how, because this is gene expression data, we can actually interrogate those drugs and we can figure out what they're doing. And in this particular case, I'm just going to focus on this cluster. And this cluster have about 4 drugs that are very distinct, and they're doing something that is very different from the other types of drugs. And in general, those drugs increase the proliferation of the cell just based on an analysis of the genes that are upregulated after the drug treatment. And you can see that there is a bunch of drugs that are generating a similar response. So in this tool, we can submit signatures and then find small molecules that can either mimic or reverse the response. In this particular, we submitted one of those signatures that we've seen, and then we can find the other drugs that induce a similar response. And then we can interrogate this for enrichment analysis for pathways, biological processes, and we can see that there's high upregulation of the cell cycle pathways and upregulation of kinases in general. And so this is an initial project that is now very close to being published, and this was 100 drugs and about 6,000 signatures, and we wanted to see the effect of more drugs. So using this fireworks type of plot, we can look at visualize many of those signatures. And here we are looking at coloring by time point. And we have 3,000 drugs, 17,000 signatures, 63 cell lines, 3 time points, and 51 doses. So this is a much larger collection, but we also filtered out all of the signatures that were insignificant. This is a coloring by the p-value, by the strength of the signature, and you can see that most of the signatures at the edges are very significant. Those are the red spots. And the most informative is the coloring by cell line. So some of the perturbations are very cell line specific, but there are some areas that you have very strong responses that have many of the cell lines aggregated in those responses. And those responses are very distinct. So we can find many chemotherapeutics in this cluster, and we find some novel drugs there that basically generate or induce the same response and those could be potential chemotherapeutics that we just don't know that they are chemotherapeutics. And the same goes with HDAC inhibitors, CDK inhibitors. For example, this small molecule is reported as a FAC inhibitor, but in reality is actually with a cluster of CDK inhibitors. So what we can say is that we suspect that this particular small molecule is indeed a CDK inhibitor. And then we have here some more downstream kinases that include CDK inhibitors, but some other downstream kinases. And then here are the EGFRs and the mTOR. So this visualization enables you to start seeing globally what happens to all of those human cell lines when you treat them with many drugs, and you get a picture of those responses, and you can find novel drugs, you can interrogate the potential functions of those drugs, and identify the mechanisms of those drugs. So I'm going to shift for another project where we try to integrate proteomics and transcriptomic data. This is RPPA data and mRNA expression from CCLE. And we cluster the data and visualize it using the TSNE method. And there are 400 cell lines that have now available transcriptomic and proteomics data. And you can see that the cell line cluster by their tissue of origin, but they also cluster by their e-coherent status or the EMT status. So the EMT is a really strong, a mechanism that influences the state of the cells across cancer types. And what we found is that there's a lot of those switches that are bimodally expressed in the EMT, e-coherent high cells, and e-coherent low cells. And just to make the story short, we identify a new EMT signature based on this combination of this data and the idea of this bimodality of those genes both at the mRNA and protein level. And now we can interrogate those with linked drugs and see which drugs would induce EMT or potentially reverse EMT. And what we find is that kinase inhibitors in general, they're reverse EMT, so they are pushing the cells toward the MET phenotype, while HDAC inhibitors are more likely to push the cells towards EMT. So what I showed so far is looking at samples, at cell lines, signatures, but we can invert the matrix and now look at genes. So this is the entire human genome, so every dot here is a human gene. And then we can start clustering the genes based on their co-expression, based on the links data. You can do that with other data. And then you can, what on the right is the same plot, but now I'm coloring the Go terms that are available, like the number of Go terms that are available for each gene. And we can see that there are many genes that don't have Go terms, so we don't really know what they're doing. So, and those genes are all aggregated in those black spots, but there are also a lot of dark black genes that we don't know about that are together with the red genes that have known function. This means that we can infer function for genes, and if we do this just for kinases, for example, we create a co-expression network of all of the human kinases, we can do this clustering analysis, and then we can zoom in into one of those clusters and see which ones of those kinases. So here we focus on a cell cycle cluster, and then we can find some kinases that are in this cluster of cell cycle that we don't really know anything about yet. So this particular kinase, PKMYT1, it's a kinase that we don't really know much about, but when we looked at TCGA, we see that it's upregulated in all cancers in TCGA when it's compared to normal. And it's also, for some cancers, it's a really good biomarker for the survival analysis. So this particular kinase could be just by itself already tell you about survivor, and because it's in the cell cycle, because it's upregulated, it's probably a good target. And now with the links data, we can ask what are the small molecules that maximally downregulate this kinase. So we just query it with this new tool that we haven't published yet called Dr. Gene Badger, and it identified this vemurafenib as a potent downregulator of this particular kinase. So this is a BRAF inhibitor that was identified by the L1000 data, but also using a collection of signatures that we processed from the gene expression omnibus by looking for studies that use this small molecule or drug and then measure gene expression before and after drug perturbation. So we also have our own collection from the public domain, and then we can compare it to the L1000 data. So we got the same drug in both searches, and that gives us confidence that this drug is probably a potent downregulator. It's not specific. It's not necessarily targeting the kinase, but it's a potent downregulator of that kinase. So in summary, I want to mention a movie that I watched about a week ago on the plane. It's called Arrival. And in this movie, the aliens, they come to Earth and they try to communicate with the humans using those images. And the people are really scared of the aliens, but they think they're going to kill us. But in actuality, they found out that the aliens want to give us a gift. And this gift is a language. And the language is those images that if we understand how to read them, we can, in the future, we can see a time, or we can see the future. So the interpretation of the images was done by two scientists in the movie. And in one hand, we are doing something similar with the figures that I showed you. Now the visualizations of data is becoming very powerful, and it's no longer those big, messy networks. We can actually visualize many nodes and perturbations and samples and actually see what is happening at the global scale. And the idea of like a language or those visualization will also change the way we think about, perturbations and cells. It's something that the LINKS program is interested in advancing or promoting, is that once we have those global views, we can really understand how to develop better drugs and accelerate the field in general.